Okay, I think we can probably get started here. Like most people have shown up. Um, so before beginning, we would like to respectfully acknowledge that the island of Newfoundland is the ancestral homelands of the Micmac and Beothuk. We also recognize the Inuit of Nunatsiavut and Nunatukovut and the Innu of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful and equitable partnerships with all people of this province as we work towards true reconciliation and honor this beautiful land together. Just want to make a quick announcement before we start that next week we'll be advertising for the Geography Blue Box seminar instead of our regular BGSA seminar. And so this should be an interesting seminar for everyone. It's going to be a remote seminar, and the speakers will discuss feminizing the environmental science policy interface in Canada. The important thing is there will be a slight time change. So instead of our usual noon seminar, it'll be from 3 to 4 p.m. on the 26th. So just keep that in mind, and we'll have more details in next week's newsletter. So today, I have the pleasure of introducing our speaker, Dr. Amanda Savoie. Amanda has a PhD from the University of New Brunswick, and she is now a phycology research scientist at the Museum of Nature in Ottawa, and a director for the Center of Arctic Knowledge and Exploration. Oops, 1 second. So today she will be giving a talk on some of her work on macroalgae. So welcome, Amanda, and you can take it away.
Thank you very much. I'll just find my slides. Okay. Hopefully that looks okay. So thank you so much. I'm really happy to be here talking today, well, virtually here anyway. And I'm going to talk a little bit about my work on seaweed diversity and distributions, biodiversity, and then also how I'm really trying to incorporate natural history collections now that I work at a museum. So that's where I've got my old and new collide. And yeah, thanks for the intro. I'm a research scientist in botany. So I work in the botany department at the Canadian Museum of Nature. I'm a phycologist, and we're a very small team at the museum. And recently also I've been directing the museum's Arctic Research Center, which means basically that I'm trying to help researchers organize Arctic research, sort of synchronize the Arctic research that's being done at the museum, and look for new opportunities too. And I fell in love with seaweeds during my undergrad at UNB, at the University of New Brunswick. I took a marine course that was the block format course and had all these different sections, coastal ecology, ornithology. And for whatever reason, I just said, okay, seaweeds, this is it. This is what I want to do. I really, really, really enjoy working on seaweeds, and I think they're so cool. So hopefully I can kind of give you a tiny little bit of understanding of why they're so interesting. And I worked at UNB for my PhD as well. And I worked on the taxonomy of a specific group of red algae that's really traditionally difficult to identify, the genus Polysiphonia. So that's what that's how I kind of got here. But now I'm working at the Canadian Museum of Nature, and I just want to kind of talk about what the museum is as well, because now all of my work is sort of starting to be framed in a different way. And the museum is Canada's National Natural History Museum. And we have, this big fancy building downtown with beautiful exhibits. But what a lot of people don't know is we also have a research department, and a really active research department that's doing work in a lot of different fields. And, most people even in Ottawa don't know this. So I'm always kind of trying to spread the word about this as well. And the mandate of the museum is to increase interest in knowledge of, appreciation, and respect for the natural world. So the idea is to accomplish this via, exhibits that people who aren't scientists can come and look at and learn something new. And then also by having a research program that's studying the natural world. And you may not know, depending on how much you know about museums, is that almost all the specimens that a museum has are not on display. So something like 1 to 2% of the specimens that we actually have are on display. And then in a research facility that we have in Gatineau, Quebec, where I work, we have 14.6 million specimens stored. And these are stored in perpetuity for all Canadians. So the idea is that they're a research collection, a collection that can be accessed by anybody who's interested in doing research on those collections. And that at any time, you could come to the museum if you wanted to look at the dinosaur fossils, if you wanted to look at the egg collection, if you wanted to look at the entomology collection. The idea is that these are here for research, for people to look at them, for studies to be done. And there's, really careful conservation of these specimens. So our mandate basically is to preserve these collections for Canadians. And it's really, really cool trying to develop new research programs that use these collections and make use of them. So I want to briefly touch on, because I'm a phycologist and a botanist, what is a herbarium? So we have the herbarium collection at the museum is the National Herbarium of Canada. And we like to call it, there are flat plants and algae as well. So this collection of flat plants was created from the plant collections of the Geological Survey of Canada. So at the very beginning of the museum, the sort of start of our museum collections was the collections of the Geological Survey of Canada in the mid 1800s or so. And then in the sort of late 1800s, early 1900s, the museum hired the first biologist, who was John McCown, and he collected all over Canada. He collected on the West Coast, on the East Coast, and I think a bit in the Arctic as well. And he was a botanist. So we actually have a really, really, really good collection of quite old specimens, of botanical specimens. And he had something like 100,000 plants by 1911 in our collections. And he also traded a lot. So I've been working in algae collection, and I find all these crazy things from England and from different places that he was very actively trading specimens with other people. So thanks to him, we have quite a good botanical collection, and some of our oldest specimens date back to 1766 and are actually collected by Joseph Banks in Newfoundland. So it's definitely, those ones are locked up in a special cabinet, but we got them. And so if you don't know, you can actually press and mount algae just like you can with plants. So with plants, you often press them in between newsprint and, they're mounted on a piece of paper, but what you can do, sticky, you can actually mount them and press them directly on a piece of paper right off the bat. So you take your collections from the field, you put them on a piece of paper, you cover them with cloth or something else so they don't stick, and dry them. And when you're done, all of your collections will be nicely mounted on paper, and they can be stored in a herbarium cabinet. So this is a really useful way to collect and then store specimens. And so a question I get asked all the time is, oh, okay, so there's research at the museum. We didn't know that. So then what kind of research happens at the museum? And the answer is, in all of these different fields, we have botany, zoology, paleontology, mineralogy. Most of the work that we're doing is collections-based research that's on taxonomy, systematics, and biodiversity. And I think this is really important because at a lot of places, a lot of universities, a lot of research institutes, taxonomy and systematics especially are really not, I think they're not valued in the way they should be, especially in terms of trying to get grants or trying to get research funding. They're not, sexy scientific disciplines. They're very but they're very much at the core of scientific research, especially things like discovering new species and how do we identify species. And so a lot of researchers at the museum and at museums in general are working on these fields. And the other main thing we're trying to do is conserve and maintain natural history collections. So the collections that I talked about, part of our job is to kind of keep them going, make sure they're in good condition, make sure they're accessible to the public, and that sort of thing. And these collections are really, really important because each collection is a record of where species occurred in space and time. So if you think of all the changes that we're seeing in the world today, climate change, biodiversity loss, species extinctions, there are things that we'll never get back. But if we have a museum record of this species or this occurrence, at least we can try to use that to make predictions about the future. And we have some interesting things there's a malacologist who works on freshwater mussels, and we have collections in our specimen, in our collections about there were mussels that were collected kind of like where Parliament Hill is now, where there's a bridge, where there's, there was no mussels growing there anymore. But it's cool to think that the Ottawa River used to have these mussels and all of this diversity, and you can kind of see what the environment used to be like before industrialization. And so these are all good reasons for conserving these specimens, for doing research on these specimens. And so I want to talk a little bit about my work, and kind of introduce algae, introduce systematics. Because again, I'm up here on my soapbox talking about systematics, but I just really want to highlight kind of how important I think it is as a research discipline. At its core, it's the study of biological diversity, and systematics is, species diversity, genetic diversity, and then also evolutionary relationships. So, thinking about how did this group evolve? How is this genus related to this other genus? How are these higher taxonomic levels related to one another? And I did spend a lot of my PhD kind of working on similar things like that, especially higher taxonomic levels. In red algae, things are still not resolved in some groups, or they're very poorly understood. And so there's actually quite a lot of work to be done to understand how different red algal families relate to one another and that sort of thing. And then as well, taxonomy is more specifically the science of naming, describing, and classifying organisms. So just good old-fashioned species discovery. And I love this quote. It's from 1985, but it still applies today. We have no idea how many species there are on Earth. There's so much work being done in biodiversity and species discovery, but at the end of the day, there's just vast worlds out there of species that, we have no idea even exist, whether in insects or in algae or in any of these understudied groups. But then even in really well-studied groups, sometimes species are still being discovered. And I think it kind of opens your mind to all the possibilities of what could be out there. And then also the sad truth is that we're probably losing species that we don't even know exist, especially in really under areas that haven't been explored, that are facing climate change and different changes. And this paper was really interesting, published I think in 2008, something like that. They were trying to sort of estimate how many species are there on Earth and in the ocean. And so they used some kind of method that looked at the rate of discovery in different groups, how commonly are species being described, and then tried to estimate based on that how much there must be still out there. And at the end of the day, it's an estimate, but they estimated that 86% of terrestrial species and 91% of marine species remain to be discovered. And that there's probably, sorry, my doorbell, there's probably 8.7 million eukaryotic species on Earth. And so this is a huge number. And, it's an estimation, and it could be right, could be wrong, but it still kind of shows you how much work there is to be done in terms of biodiversity, how important it is for us to kind of at least attempt to kind of describe and assess the biodiversity on Earth. And then I won't even get into it because this estimate doesn't even include prokaryotes. So this is only for eukaryotic life. And so, the diversity of bacteria is even above and beyond this. And the marine environment clearly has more need of biodiversity work, of species diversity work than the terrestrial. I mean, clearly they both need work, but the ocean is such a vast, unexplored habitat. We've barely scratched the surface of exploring the ocean. And I think even though the coastal zone maybe is somewhat relatively well described, there are still whole new habitats being discovered in the deep sea, species with wild species that we never could have imagined. So anyways, it's very, very interesting to think about what we know and don't know about the Earth. And so for me, you can't study everything, even though it's all interesting. But for me, my main focus is on marine macroalgae. And in a really general kind of sense, the goal of my work is to focus on how many species are in Canada and then where do they live. So obviously oversimplifying, and obviously I'm focusing on certain groups as I can, but That's sort of the general idea behind what I'm hoping to accomplish. And so, what are algae? And this is a really interesting, you know, it's a really interesting thing to talk about because a lot of people don't know that algae, the term algae is not a monophyletic group. Um, you know, commonly people were taught that algae are protists, but protists are also not a monophyletic group. And it's not really it's not like an evolutionary lineage or a meaningful natural group. So it's just a term that is convenient for us to use to discuss photosynthetic organisms that are not land plants. So basically algae are photosynthetic. They usually live in water or need water somehow, but they're not land plants. So that's how they're defined. So this quote from an algae textbook is that algae are a heterogeneous assemblage of organisms that are largely defined by ecological traits. So basically, it's sort of just this grouping of everything that photosynthesizes that isn't a vascular plant or a bryophyte or liverwort. So there's a wild, as you can imagine in that case, there's a wild diversity of algae. There's more than 35,000 species. It would take a lifetime to even understand the kind of diversity within this group. And I've mentioned a few times that I'm a phycologist. And so the scientific study of algae is called phycology. And you will get autocorrected by Google every time you try to search for the Journal of Phycology. But I think my computer has learned by now that I don't want to look up psychology. So phycology is from phycos, from Greek for seaweed. And you can see here, I don't know how to get a laser pointer, but if you look at the right of my slide, you can see this sort of diagram of the different eukaryotic supergroups. And there's 6 eukaryotic supergroups, and 4 of them have photosynthetic organisms. So algae are really widely distributed throughout the tree of life. So within Plantae, this is the group that, we have land plants. We also have red glaucophyte and green algae. And then brown algae are found in the Chromalveolate group. And then the other groups also have different types of photosynthetic organisms. And so they're not related to one another. They're very, very distantly related to one another in some cases. And at the bottom, there's just some examples of different unicellular types of algae. And so that's algae as a huge kind of overarching group. So then what are seaweeds? Seaweeds are in the picture here. They are a subset of algae. So when we're talking about seaweeds, we're talking about algae that are basically macroscopic and marine. So again, seaweeds is not a phylogenetic group. It's not an evolutionary lineage. It's just a convenient grouping for scientists to kind of talk about a subset of organisms that share characteristics. So seaweeds they are usually found in coastal regions around the world. They're basically any algae that we're talking about that's macroscopic and almost always multicellular. There are a few weird green algae that are one large single cell. They're like 20 centimeters long and they're somehow still a single cell. But in the most cases, they're multicellular. And the main kind of determining factor for where you can find seaweeds is that they need some kind of substrate. So they almost always need to be attached to something. There's occasional habitats around the world where you can find seaweed, like the Sargasso Sea. There's free living seaweed, but in most cases, it needs some kind of substrate to attach to. So they're usually found on rocks, the rocky intertidal. They can be found growing on coral. They can grow, and very commonly do grow on each other, and sometimes very specific relationships, like this one only grows on this one. And then we also find them on animals. So there's, on crabs, like decorator crabs, that kind of thing. They will also have algae growing on them. And then there's also some species that grow on sea turtles and then also on manatees. So it's kind of an interesting habitat for seaweed to grow. But generally speaking, we're talking about finding them on rocks. And so you won't find them on sandy beaches. Like you won't find them growing in the sand. You might see things washed up from further offshore where there is rock, and you won't find them growing on cobble. So the cobble would be turning too much, and any algae that try to grow would get scoured off. And so there's 3 groups of algae, of seaweeds, and they are red algae, brown algae, and green algae. So red algae are often found in the lower intertidal. They are a phylogenetic, they're a monophyletic group. Same with brown algae. Brown algae are found throughout the intertidal zone, but there's a really high biomass of brown algae, usually in the middle intertidal zone. And brown algae are probably, most people are most familiar with brown algae because they know what kelp are. So kelp are brown algae, and they're like big, they're kind of like the charismatic mega flora of seaweeds. They're the one that most people know about. And then green algae are the group Chlorophyta. So green algae, you might have seen sea lettuce or other things like that that are green algae. And seaweeds are really important. They're primary producers. Obviously, they're photosynthetic organisms. So they're really forming the base of the food chain in the coastal zone. You always see snails and all kinds of things eating seaweeds. There's often urchins eating seaweeds. So they're supporting these higher trophic levels. They also have a really important feature in that they kind of add this structure to the intertidal zone. So if you've ever been out on the intertidal and kind of digging around through seaweed, what you'll find is there's all kinds of organisms living in and on the seaweed. So as the tide goes out, the intertidal zone without seaweed, we'd be totally dried out from the sun. But seaweeds have a lot of adaptations to hold water in their bodies. And so all the other little organisms, invertebrates and things hide in and on the seaweeds, and that way they don't dry out either. So they really provide a habitat. And then seaweeds also they often shade a tide pool. So if you have a tide pool that has seaweed kind of covering the surface of it, then that makes a better habitat for small fish and crabs because it's shaded, it's protected. It's not going to experience temperature extremes to the same level. And then also there's been a lot of research recently about how kelp and large seaweeds are a carbon sink. And so I just wanted to show a beautiful picture of a site I've collected a lot in southern New Brunswick in the Bay of Fundy. And so seaweeds are basically found in the intertidal and the shallow subtidal all around the world. So there's kind of a narrow strip around the coastline where seaweeds can be found. Because they're photosynthetic, they can't live very deep. So the more clear the water is, the deeper they can be found. But there's limitations, of course, because if there's any silt in the water or if there's too much water movement, you won't find them too deep. So in New Brunswick, they're not as deep as they might be off the south shore of Nova Scotia, where there's a little bit less mixing. And so I think I'm trying to think maybe 80 feet, 90 feet, you're kind of running into the end of where you see seaweeds. But in the tropics, they would be found much deeper than that because the water is so clear and there's more light coming through. But in the Bay of Fundy or anywhere that has a really high tidal amplitude, you're going to have a much higher biomass of seaweeds because there's so much more real estate essentially for them to take up in the intertidal zone. And then overall, Canada is an amazing country to be studying seaweed or anything in the coast because Canada has the longest coastline in the world. And so the coastline of Canada is about 243,000 kilometers long. And pretty much all of that habitat, as long as there's suitable substrate, supports seaweeds. So we have an amazing diversity of seaweeds on the West Coast. We have a pretty high diversity on the East Coast, although it's less than the West. And then actually in the Arctic, there's an amazing amount of seaweeds as well. There's less species diversity, but there's really, really high biomass. And it is really shocking that kelp can grow even in the high Arctic. I've seen video from northern Greenland, up near Ellesmere Island, where there would be ice for half the year. It's dark for half the year, and there's a kelp forest growing under the ice. And so they've really adapted to all kinds of different extremes of different environments. And shorelines are among the most productive of all natural habitats. So this is one of my kind of arguments for we really need to understand what we are talking about when we look at seaweeds on Canada's coastline. They are a really, really productive and important habitat, and seaweeds are the most conspicuous primary producers on these shores. And we still have so much to learn about seaweeds in Canada. And so why don't we know everything about seaweeds? And one of the reasons is that algae are really, really difficult to identify. So if anyone's ever taken an intro seaweed course, you've probably struggled with a key, and identifying especially red algae, but also green and brown algae is really, really, really tricky. And they're harder to identify than, land plants or other organisms because there's a few reasons, and I'll go through them. And the first is they really just lack features. So a lot of seaweeds, most seaweeds really don't have much differentiation within their body plan. They're not like plants. They don't have stems and leaves and roots and all these different features. They're really simple organisms. So something like this, actually this is nori, a species similar to nori that we wrap sushi in. It's a very thin blade. It's one cell layer thick, and that's it. So you just don't have many features to work with. Okay, it's one cell layer thick and it's this big, and the cells all look the same. And so it becomes really hard to separate species when you don't have any characters to work with. The second problem that makes seaweeds really hard to identify is convergent evolution. So species look the same because they're living in the same type of habitat, facing the same kind of challenges, but they're not related at all. So we have things in two different orders. So you would think, if you think about orders of plants or animals, it doesn't take an expert to tell apart different orders. But with red algae, you end up with things that look basically the same. If you pick them up on the beach, you wouldn't be able to tell them apart necessarily, but they're in different orders. And it's not until you cut them up and look under the microscope that you can figure out what they are. And then finally, there's also a lot of phenotypic plasticity. So this is a species on the West Coast, Maziella splendens, and if it grows in certain type of water, high, fast-moving water, it'll look different than if it grows in slow water, and to the degree that people would classify it as a different species if you were just using morphology to describe species. And so you run into all kinds of issues of confusing different species and not knowing how many species there are and what are these things supposed to look like. And in the past, we had, species were described based on the information they had at hand, which is morphological characteristics. The other issue that I won't even really get into is that red algae and some browns have really, really, really convoluted and complicated life histories, where one life stage can look completely different than the other life stage, to the point that they're classified as different species until somebody figured out that they were the same thing. So basically, we have run into like all of these issues, and using taxonomic keys with seaweeds also requires a high level of expertise, because the features that we're using are so technical, because the simple morphology of the plants, the things that you have to use to identify them end up being really technical, like reproductive features, for example. So basically, overall in Canada and around the world, we have kind of overlooked species diversity, incomplete biogeographical information, distributions are not well known. Complexes of species are commonly identified as a single species, and true algal biodiversity overall has been underestimated historically. And so the answer to some of that the lab that I did my PhD in was working on was using DNA barcoding to try and help solve some of these issues. And so DNA barcoding uses a small standardized piece of DNA. So within different groups, there's a different DNA barcode. There's no one kind of universal DNA barcode for all of life. But in red and brown algae, we use a mitochondrial marker it's called cytochrome oxidase subunit 1, and we're using that marker for red and brown algae. And then for green algae, TUF-A or RBCL, they're plastid markers. And so basically what these allow you to do is collect a whole bunch of specimens, sequence them, and say, how did these specimens, how are they sorted if I sort them by their DNA? Which ones have the DNA that's similar and which ones have DNA that's different? And the thing is that this can be incredibly helpful. And you don't use it alone, you use it in combination with morphological assessments, but it can be incredibly illuminating. And it can help you, what it can do as well is it can kind of help you to sort your collections and say, I think these two are different. I see some morphological differences. Then you sequence them, you're like, oh, and the DNA is different. Okay, all right, let me examine this more closely. It's because there's definitely something going on here. So it can be kind of like a tool to help highlight differences between groups. It can be used for just identification, for species survey, but every time we do a species survey on seaweeds using DNA barcoding, we find new species, essentially, either overlooked species or introduced species that hadn't been discovered yet. And then the other thing is that training taxonomists is really, really expensive and difficult, and basically almost never happens anymore. And DNA barcoding is really, really quick, easy, and inexpensive. And it's every day it's getting cheaper. And I know that there's all these other new ways of doing like e-barcoding and meta barcoding, but at the end of the day, we still need to do the very basic kind of DNA barcoding with Sanger sequencing in order to establish our barcode library. Someday, when the barcode library has been well established for seaweeds, the way it has been for some other groups, then it'll be even easier to identify species. But right now, we're still at a point where we need taxonomists to confirm those identifications that are done with DNA barcoding. And yeah, so like I was saying, seaweed life stages can be really complicated. And so one good example is a species called Von Masonia hemifera, which on the top is the gametophyte and then the sporophytes on the bottom. And so they look completely different in their different life stages and were originally classified as two different species. And now it doesn't matter which life stage, if you sample something for sequencing, if you misidentify it because it's at a confusing life stage, your DNA barcoding will tell you pretty quickly that that was a mistake. And then you can go back and confirm your work. Or if you have a fragment of a specimen or something that just isn't working, like you can't identify it, then DNA barcoding is really, really helpful for that. And so I want to talk a tiny bit about red algae and show a little bit of the work that I did, just a very small highlight of some of the work that I did in red algae. And red algae, like I mentioned, are a distinct eukaryotic lineage. They're closely related to glaucophytes and green algae and land plants. They're sort of in that group, but they're unique because they lack flagella and centrioles at all phases of their life history. So red algae, their gametes do not have flagella. They're non-motile. It's led to all this kind of crazy adaptations that they have in their reproductive patterns, and they're very complicated, so I won't get into them now. And then the red color they have is due to these accessory pigments, phycoerythrin and phycocyanin. So that's kind of the defining features of that group. And red algae are mostly marine, highly diverse, and there's about 7,000 species worldwide. Probably more than that, to be honest. But that's about the estimate when you look it up that people are saying. And then for my PhD and for my continuing work, I really focus on a family called the Rhodomelaceae. So this is the largest red algal family. There's more than 1,000 species in this family. It's desperately in need of modern taxonomic assessments, and all the groups within this family have traditionally been difficult for morphological taxonomy. Taxonomists use certain genera within this kind of almost like a dumping ground. Oh, we can't tell what it is, put it in there, let's just walk away. And so I was happy to be able to clean up kind of one tiny little part of this group and some of my work, and sort of learn to be able to identify them as well. And so there are 16 genera and 51 species in Canada in this family. And they generally have the structure I'm showing on the right. When you take a cross section, they look like little wagon wheels because of the structure of the cells and the main axis. And these are species that you would commonly see on the East Coast. If you were in the intertidal zone in Newfoundland, there would be species within this group. And basically, just briefly, go out, we collect, press everything that you bring back, and then take a small subsample for DNA. And so the idea is you always have a voucher specimen that is linked to a unique DNA sequence. So the idea is that you don't ever want to be mixed up about where your DNA sequence came from and which specimen. So I don't press multiple samples on one sheet, and I don't, some people collect duplicates, and then they send them off to different herbaria. And I would do that with species that I was confident about. But when you're working with things that are tricky and you're not sure if they're the same thing or if they're different, then every unique specimen is it a unique specimen and has its own DNA sequence and its own collecting number. So just trying to avoid the situation we've seen in the past where we have multiple red algae on a herbarium sheet, and we look at them and say, oh, there's 4 different species on this sheet. So generally trying to avoid that issue from happening. And then I'm just showing you this. You don't actually have to look at this in detail to show how we can use molecular data to kind of answer questions about how things are related. So I did a huge survey of a bunch of species within this family, the Rhodomelaceae, and I did 2 different genes, CO1 and RBCL, and then used phylogenetic analyses to kind of look at relatedness. But I'm not really going to talk about that right now. I just wanted to show you kind of the power of being able to sequence all of these groups. The main thing I want to highlight is a couple of new species that I found just from doing a survey in British Columbia, and how you can use barcoding to kind of really quickly and easily highlight new species. And then the main thing I want to focus on is that then that it becomes a tool to help actually find morphological characters as well, to identify them. So I'm going to focus on 2 species that I found that wouldn't have realized they were different without DNA barcoding. But once we found this group, then we were able to say, oh, look, there is actually morphological differences here too. So this species was really closely related to another species called Symphyokladia dendroidea, but it's different because it's much smaller. And this grows in little clumps in the subtidal to about a depth of 18 meters, but also can be found in the low intertidal. And we found this one on Vancouver Island and in Haida Gwaii. And basically, I wouldn't have noticed that it was different because it just looked like a smaller version. Oh, it's a little bit smaller than the other one. But it had a very distinct barcode, and this was consistent among different markers. And then once you sort your samples using barcoding, then you can kind of sit down and say, oh yeah, okay, there are some differences here compared to different other specimens, and try to figure out, okay, this is actually a new species and it's consistently different among all the collections we have. And then there was another one called Symphyokladia rosea that was also different. And this one was interesting because it was only in the subtitle. It was found in British Columbia generally and in California. And then this one was even more different. It was, this one I would say was more just overlooked, probably because it was in the subtitle, which, not everybody's collecting while scuba diving. And so this one, at the end of the day, after sorting through everything based on the barcode results, there was a distinct pattern of branching and the size of the specimens that led it to be able to be differentiated from this species that it was closest to. And so I kind of wanted to highlight that a lot of people talk about cryptic species with DNA barcoding, and we do find cryptic species. But a lot of the times what we're finding is species that do have morphological differences, but they're not distinct enough that you'd immediately say, wow, that's something new. It's more like once you sit down and look at it, because you've had this sort of help from the DNA telling you there's something going on here, then you can distinguish them. And so it's a really powerful tool. But what about museum specimens? So now I kind of been trying to wrap my head around, how can I use this, we have an immense database of DNA barcode specimens, but we have all these historic museum specimens that aren't barcoded, and in some cases can't be barcoded. And so it's interesting because I'm now trying to wrap my head around, okay, I have this amazing technique that can be really helpful for studying red algae and for studying species diversity, but I also really, really want to use valuable historic museum specimens in a meaningful way. But I have to acknowledge that some red algae are really hard to identify without being able to sequence them. And so it's kind of like, it's something I've been thinking about over the last 2 years that I've been at the museum, is how can we kind of keep moving forward using molecular techniques and eDNA and all these powerful tools, but also really have really valuable collections that in some cases can't be sequenced. And I really want to be able to use them for biodiversity research. And so there's kind of 2 approaches. And one is barcoding historic specimens. So this can be done. If you really want to try and sequence a historical specimen, you can, but it's expensive, it's difficult, it requires a special, in some cases you need a special ancient DNA lab. I think it is useful in the sense that there's quite a few people that have been trying to barcode type specimens in algae. So they take the specimen that the whole species description is based on, the type specimen, and they really work to sequence that. And you get kind of fragmented sequence. You don't get great quality DNA, but you often can get something. And in some cases, it can be helpful. But I don't think that this is the approach to go on a wider scale. Like, I think it's more useful if you're like, I really, really need to know what this one specimen is, but it's not something you're going to do with 1,000 specimens in a herbarium collection. And it's not something that would be kind of cost effective in the same way that DNA barcoding is. And in some cases, it's not just that herbarium specimens are old. DNA degrades quite quickly over time, but also that it was really common, and this was the case at the Canadian Museum of Nature, is that the phycologist who was working there in the 60s and 70s would pack all of his specimens in formaldehyde before pressing them. So he would be collecting in the Arctic, and then he would ship everything in barrels in formalin and then press them afterwards. And so now we've had this extra obstacle. We have old specimens that were preserved in formaldehyde. So realistically, getting DNA out of those would be it's probably possible, but would be a challenge. And so I think the best way to kind of make use of herbarium specimens is to learn the most we can about the biodiversity in Canada using modern collections, and then take that knowledge and try to apply it to herbarium specimens. So the example I just gave is that I kind of learned something about this genus Symphyocladia because I barcoded the specimens. But then once I barcoded them and really assessed them carefully, I realized, oh, there is a way I can tell them apart. So what I'd like to do is kind of apply that, then to take that kind of knowledge and apply it to herbarium specimens. So if we can have really clear, and it wouldn't work in every group because there's some groups that they're too difficult to identify. But if we can kind of take the knowledge that we gain from modern assessments of DNA barcoding groups and apply them to historic collections and say, okay, maybe I can tell these apart if I really sit down and work on them carefully, then we're able to use these herbarium specimens to kind of make predictions about the future and look at how things have changed compared to even in the last 50 years. So groups that are easier to identify and well studied should work well. And I was really happy to participate in a study and publish a paper looking at this kind of thing with kelp. So I was working with a group based out of DFO and Université Laval to try to predict kelp distribution with climate change. Essentially, how will kelp distribution change with changing climate? And they had some of their own collections, modern collections, but they wanted to also incorporate museum specimens. And so what we had to do is we had to sit down and say, which kelp species can we include that we can be confident we can identify them based on a picture or looking at a specimen? And so basically what we had to do is kind of sit down and say, okay, we can't include Laminaria digitata because we can't tell Laminaria digitata and Hedophyllum nigripes apart without DNA. But all of these other species, we can tell them apart without DNA. So let's use these ones. And so it was really cool to be able to combine kind of modern observations, historical observations, some of them over 100 years old, of kelp specimens, and then have the modeling people make a model to try to predict, okay, so how is kelp distribution going to change over the next 50 years if climate change and sort of worst case scenarios for climate change. And what we saw was somewhat predictable in that Saccharina latissima, the sugar kelp, is probably going to gain more habitat, and Longicruis, the other Arctic kelp, is going to lose habitat. And so species that are dependent on cold water will lose their habitat, and species that are more tolerant will gain habitat. And so what I would really like to do is to do more studies like this, but with red algae and with our Arctic collections. So the museum, and I'm also sharing this because I'm hopeful that other people might say, oh, wow, I want to do museum studies too. And a lot of people don't know we have these collections, so I'm also just trying to get the word out. We have really cool historic collections from the Canadian Arctic. So we have collections of seaweed from the Canadian Arctic expedition that was done in 1913 to 1918. And so think of how much the Arctic has changed since 100 years ago. And then we also have collections from Lee, who was the phycologist at the museum in the 60s and 70s. And Lee collected so many species. So these are from the Canadian Arctic expedition. And then we also have these surveys that he did. It's actually amazing. And I wish I could find more information about it. I need to go to the archives and kind of talk to them. But he had amazing helicopter support. He was all over the Western Arctic in remote, isolated areas, scuba diving under the ice in the 1960s without a dry suit. Like it's totally wild. And he collected so many specimens. And we have all these specimens at the museum. And what I want to do is find a way to compare the seaweed distribution in the Arctic today with global warming occurring and in the past from the 60s, and then also try based on the small amount of specimens we have from 100 years ago. And so these collections are going to be really, really useful as long as I can find, and I should be able to find groups that are relatively easy to identify and can kind of do this type of study that I would like to do. And so what I want to do is kind of use these collections to try and help predict future changes in seaweed distributions. And then the other thing that I'm working towards, and that the museum is working towards, is how can museum specimens in general be made more accessible for scientific study? So one of the biggest ways we're doing this right now is digitization. So instead of having specimens, basically, technically they're accessible to the public, but in practice, they're locked in a herbarium cabinet in Ottawa, right? But if we can have everything digitized and available online where you can search and say, I want to see all of the phycoteries from the Canadian Arctic, and you can see all of our collections sitting at your computer, that's the ideal situation. And that's something we're working towards. And then this data would be available to everybody. And so in the future, the project I'm working on right now is I'm hopefully going to be going to Cambridge Bay in the Canadian Arctic this summer. That's the new Canadian High Arctic Research Station is built in there in Cambridge Bay. And they're really keen to do a seaweed survey. And so what I'd really like to do, well, I have so many things I want to do, but I want to first go and engage with Indigenous people at Inuit in the area and talk to them about their experiences with seaweed, what they know about seaweed in the area, and try to incorporate some of that knowledge into the study. And then also I'd like to go and find sites where Lee collected seaweeds and see if I can go collect there as well and try to compare directly to historical collections and try to see what has changed. And then the other main thing I'm working on is getting all of those collections that I make and historical collections to be digitized and be available online. So that's the goal. We'll see. It's all a work in progress, but I'm really excited about the possibilities for all of these connections. And basically, I just want to finish by saying that, kind of saying again how critical museum collections are for conducting kind of baseline biodiversity science. That historical collections have often been overlooked, but they're a really important source of baseline data. That going forward, all the new collections I make will also, and I didn't even touch on this, all the new collections I make will have DNA material saved. The museum has a cryobank, which is basically a set of freezers that use liquid nitrogen to keep samples frozen at like minus 160 degrees Celsius, something like that. So everything we collect going forward will have a tissue sample frozen in liquid nitrogen. And that tissue sample is also available to anybody who wants some of it in order to do research. And so going forward, museum collections will become even more useful because they're going to have this genetic material associated with them. And then finally, I'm looking forward to when, all museums are able to digitize their collections. And we're going to have all of this information about biodiversity available and that anybody can access from their computer. So I think that's about it. And thanks for listening.
Great. Excellent. Thank you, Amanda. That was a great talk.
Thank you.
I think So if everyone can just enter any questions that you have in the chat, then either I or Amanda can read them out, and we'll just go in order that way. So feel free to do that whenever you're ready. And when you ask your question, you can unmute your mic and turn on your camera if you wish. Okay, we'll start off with Yolanda. Go ahead.
Hi there. Hey.
Hi.
Great. And I really enjoyed it. I was curious about just, given this is a challenging group to work with, how do you deal with mapping distribution from collections? Because it seems to me these things, they come loose, they move around, and also where and when you sample them is pretty limited, and there's probably a limited amount of experts in seaweed. So how do you translate, your collection expeditions to, range maps or understandings of distribution in a broader sense?
Yeah, it's difficult. So it's basically all those issues you mentioned. We have all of them, and one thing, I didn't even talk about is drift. So if you collect something that isn't attached, you can't say anything about where it came from. So sometimes we're like, oh, we found this thing, it's super interesting and rare, but it's not attached, and so like who knows where it came from. So usually for trying to really be accurate with distributions, I would talk about known distribution based on barcoded specimens that were collected, attached, using it, but the issue is when we look at historical lists and historical distributions, it's just all over the place. So the answer is, like, in terms of historical data, it's really poorly known, the distributions, and they're kind of guesses. And then anything that has had more modern work done has a bit more detail. And we just try to map, okay, this is identified by a taxonomist, it was barcoded, and it was definitely attached in this spot. And then, for sure it was there, at least in that point in time.
Cool, thanks.
No problem.
All right, next question we have from Kay and Taya. You can go ahead and ask your question if you want, or I can read it out if you prefer. Okay, maybe I'll just read it out then. So they ask, can you tell us any more about how the DNA sequencing process works?
Sure. Yeah, I can talk about, so let me think where to start. So basically I would collect a specimen, and red algae, and I'm mostly talking about reds here because that's what I'm most familiar with, they often grow like tangled with each other. So when I'm sampling for DNA, I'll try to be really careful and like really carefully sort it out and clean it, so that just, kind of looking, make sure that I don't have two things growing together. And then what the best way to do it is to, or at least for algae, is to take a small sample, dry it off, and put it in silica gel. So if you've ever dried flowers, or, you can buy like the silica powder at Michael's, and that's basically what we use. It's just it sucks all the moisture out of your specimen really quickly. And so you can also freeze them in a -80 freezer, but if you're in the field, you probably don't have access to a good enough freezer. So silica is something that it's like pretty easy to transport and quick and easy to dry things quickly. And then after that, you extract the DNA using an extraction process for red algae. Each group has a different extraction process. But red algae have really gooey cell walls, so it's not too hard to get DNA out of them. Then you PCR amplify, and then you sequence using primers. I won't go into too much detail, but basically for each kind of group of organisms you're working with, you need specific primers. And then if you get amplification, then you send it away for sequencing, and you get back your DNA sequences. And then there's different ways that you can analyze them. And then I use Bold. So the Bold Systems website, which was run by the team in Guelph, is really useful. You can search. So you put it in, it's like a search engine, and it pops up and it says like, it could be this. And then you sit down and say, okay, is it that, or what's going on here? Yeah.
Great. So next we have Olga, and you can go ahead and ask your question if you want, or I can read it out again. It's up to you. Okay, perhaps I'll read it out again. Oh, Okay, great, sure. Guess everyone's in a loud space. So they say thanks for the talk. I have two questions. One, how old can red algae specimens to be used for DNA barcoding, minus the ones preserved in formaldehyde? And two, would you be able to barcode all the specimens in your collection if you had the resources? Oh, sorry, that's all one question. Second question, do pressed algae specimens have any kind of smell?
Oh, so yeah, they do kind of smell a little bit like seaweed. Like you do get a little bit of a, but it's pretty, they kind of, the whole herbarium, like the plant herbarium in general and seaweed herbarium has like a little bit of a sort of, I don't know, it's like a museum smell, like a library smell, I guess. It's lots of paper and dried things. So, I don't know. It's a hard question. Like the amount of resources, because so many of the collections are really old, I don't think it would be worth, like, yes, if someone invented a way that you could get DNA out of specimens that were preserved in formaldehyde quickly and easily, I would say let's do the whole thing. That would be awesome. But realistically, it would be really difficult. I don't know. Like if something's properly dried and it's like 10 years old, I don't think you'll have an issue getting DNA out of it. It's sort of after that, like 10, 20, 30 years, you're starting to get worse. And then by the time it's like 100 years old, what happens is you have to use really sensitive primers and really sensitive kind of procedures to pick up the DNA that's in it. And then what happens is if there's any kind of modern DNA in that sample, that's going to get picked up first. So one of my colleagues in my lab at UNV was trying to sequence. We were kind of experimenting with sequencing old things, and he ended up sequencing himself, probably because he accidentally touched something, you know. And so it gets really hard to be really specific with what you're sequencing. And I am not there's people that, you know, run ancient DNA labs that are experts, and I'm not an expert in that, so I'm not sure exactly how difficult it is. I just know it's difficult.
All right, next question, I believe, is Julissa. So you can unmute if you wish.
Hello, very nice talk, Amanda. Thank you so much. It's a quick question. Where do you start the digitization process? Were there any specific specimens that received priority?
So for us, for the museum, we received donations that helped us to kind of kickstart our digitization. And so we started with Arctic because that was the donor's interest. And I think because the Arctic is kind of one of the most important in terms of climate change, important areas. And then because the museum also has a lot of Arctic specimens, it was kind of a natural focus for us. So the main priority was Arctic, but there's other smaller priorities where anything that was extremely old or historic or of like public interest was digitized first. And then type specimens were also all digitized, at least for vascular plants. That was one of the first things that was digitized because that's what researchers or students are most likely to be looking for, is looking for images of type specimens.
Thank you.
You're welcome.
Okay, we also have a question from Joseph, and if you want, you can unmute and ask your question, or I can read it out loud if you prefer. Okay, maybe I will read it out again. Oh, okay, great. I think everyone's in the grad student offices probably can't talk. So they say, thanks for the talk. You made mention that 86% of terrestrial species and 91% of marine species remain to be discovered. Is there any research going on to increase the discovery pool that you are aware of?
Yeah, I think it's a good question. So even like my own work, anyone who's doing any kind of surveying, I would say is working towards discovering new species. So for example, at the museum, I'll just talk about what I know going on at the museum. You know, there's a big botany team that goes out and surveys, you know, new areas of areas that have never had a botanical survey before. And so sometimes you find something, sometimes you don't. But at least, when you're kind of looking, and I have another colleague who works on lichens who's finding new things all the time, because lichens are another sort of overlooked organism. I think like seaweeds, there's not a ton of experts out there. So, and then we have another colleague who works on, I think the more specialized you are too, sometimes the more you're seeing things that other people don't see. So one coworker works on weevils, and he's always finding new weevil species from Central America. I mean, it seems like every other day he's got a new species. So yeah, so people are working on it, but it's one of those things that often it's like you go out and do a survey, maybe you don't find anything, but then maybe another year you do, and in the ocean it's harder for sure.
Okay, great. I think we're all caught up here. Do we have any more questions from folks in the audience? Feel free to say so. If not, in the meantime, I have a quick question for you too.
Yeah.
Kind of like a bigger picture question. You talked about like there being, I guess, or I got the sense anyway from when you talked that there fewer like maybe young scientists going into systematics and taxonomy and that kind of thing. Do you think that there's a role for like, or do you incorporate at all like any sort of citizen science? And like, because it seems like there are a lot more like naturalists, like birders and
Yeah.
like, hobbyists that are really interested in doing that kind of stuff that maybe scientists are not
Yeah.
going into that much anymore.
I think it's a combination of factors of like why there isn't as many is often it's because the funding agencies determine what science gets done, right? And so often I think it's harder to get grants that are like, hey, I'm going to go survey a bunch of places and maybe I'll find new species. You know what I mean? But I think you're right that it's not due to lack of interest, and that there's a lot of people kind of have different projects going on, and then they're kind of doing more species discovery or species surveying on the side. But yes, citizen science or community science is really important. And we had, due to the pandemic, we had to stop it because we had lost a few staff members, but we had a citizen science program going on through Zooniverse. I don't know if you've heard of Zooniverse. It's really cool. If anyone hasn't heard of it, look it up. There's all kinds of community science projects on there. And it's things like, look at all these pictures of stars and point out when this light appears, or watch these videos of gorillas and click every time you see them do this one activity. So it's kind of like crowdsourcing for scientists. So it's really cool because there's projects for every interest. And what we had a transcription thing where we put up images of Arctic plants and lichens, and then people could type what they see, the species name, the date, the collector, and that information would then basically go in a database, and we could use it to populate our database. And how it works is I think every specimen has to be done 3 times. So that is, if someone makes a mistake, that mistake doesn't get put into the database. But yeah, and people loved it. They loved it. And it was kind of running during part of the pandemic. And there's some people that were like, you can see like how the different users like, and so you could see like some people did it once or twice and some people were doing it every day. So it was cool. Yeah.
That is really cool. Yeah, it seems like such a like a, or maybe it's nice that they're finding ways to take advantage of that, because it seems like such an untapped resource. There are so many people that are probably so interested in.
Yeah,
like,
it's so
don't
true.
get the opportunity.
Yeah, and
Cool.
we're really lucky at the museum to have a lot of volunteers, and it's been sad because of the pandemic. We haven't been able to have them, they haven't been allowed in the building, but starting December 1st, they're allowed back in the building. So it's great because people are really passionate about all kinds of things. So yeah.
Great, thank you. Oh yeah, and Julissa just put a link in the chat there, I think, for the Canadian Botanical Association.
Oh, cool.
If you folks want to check that out.
Yeah, sorry, the first link, I don't know what happened, my mistake. It's the second one, CBA, Canadian Botanical Association, offering awards for students or any professional, any career level who would like to do field work in the North.
That's awesome. Yeah, it's the museum's been doing research in the North for quite a while, and it's a really important place to work, but it's really expensive, and especially for students to do that kind of work. So any kind of support is helpful, for sure.
We will just give folks a couple more minutes to.
Okay.
in case you have questions, but it looks like. Let's see.
I used
Anyone-
to, when I was at UNB, I used to help organize the grad student seminars. So anyway, it feels great to like come and give talks. And yeah.
We really appreciate having you. This is a great, this was a very cool talk. Thank you for joining us. I guess if that's it for questions, then maybe we'll just end it here. And thank you again for joining us. Great.
Thank you so much for the invitation. It was great. Thanks for all your questions.
No problem.
Bye. Thank you.
Bye.